I believe it is. Which I gotta go look at the calendar. I gotta go look at the calendar. Okay. All right. So I posted for y'all a couple of links, and I want to show you real quick. So if you go to our class, come on down. Okay. In chapter 6, sorry, because we're going to finish chapter 6 today. There's a crash course that's really good on actin and muscles. It's a really good video. It doesn't quite go into the detail that we're going to go into, but it's really good. So I put that for y'all. We're not watching it in class though, but I put it for y'all. And then and then I wanted to pull up, because there is an animation I'm going to show you in class today. So I really want to know where I'm going. Would be nice. Here. It's a Monday, right? Okay, so we're in chapter 6. Molecular animations. There we go, muscle contraction. That's the one we want. Okay, now we can really start. All right, I'm gonna go all the way down because we're on the very last section on my favorite protein ever on the entire planet ever is actin. Okay, so let's see if I can do this. But before we do actin, and you've done your attendance, right? Everybody's done their attendance. Please do your attendance if you have not done your attendance. Before we do that, start with a joke. So why did the football coach go to the bank? To get his quarterback. Okay. Do what? Y'all get the same joke as Charles. I am not doing double time. No, that was today's joke. That was today's joke. Did I tell y'all the St. Patty's Day joke? Okay, then I'm saving it for Wednesday. Isn't it Wednesday? So I'll see y'all on Wednesday. Okay, remind me for the extra St. Patty's joke on Wednesday. All right, so actin and myosin. Actin and myosin are what allow our muscles to contract, right? They are the protein interactions that occur that make our muscles contract. It has been an ongoing thing to understand how this works. People have wanted to know this for so long, right? And the initial models were put out. It's called the sliding filament model in 1950. I think it was 1954. There was one group in Cambridge who was working on it, one group at MIT that was working on it, and they both kind of came out about the same time. And so this is kind of, we're going to go a general. This is kind of how it works, and then we're going to go into lots of detail, right? So the main part of all of your muscle tissue is composed of two proteins, actin and myosin. The myosin are the thick filaments, so anytime you see them in a drawing, they'll look thicker. Actin filaments are the thin filaments. So there are these little head groups on myosin that actually bind to the actin. And when you think, I would like to contract my arm, right? And your arm comes inward. What happens to the length of this muscle? It gets shorter. Now, if I were anybody else, when I would do this, what would you see my muscle do? If it were Arnold Schwarzenegger, what would you see my muscle do? Right? It would make a big bulge. Not mine, but Arnold it would, right? So what's happening to that muscle? It's going from very long and thin to very short and fat, right? That's what making a muscle is, right? Okay, so how does that translate inside the cell? Well, when you contract, what you're actually doing is these myosin head groups are grabbing onto a position on actin and then basically pulling them to the center, letting go, doing it again and pulling. And so like the ends would be the ends of the classroom, right? And when we would pull in the sides of the classroom, the height of the classroom would get taller, right? Okay, so that's kind of how muscles work. So we call this from Z disc to Z disc, we call this one sarcomere. So here, nope, come on. So from here to here is one sarcomere. So Z disc is like the end of the sarcomere, Z disc is the end of the sarcomere. So look at the total width of the sarcomere. Compare contracted down here versus relaxed up here. What's different? The width, right? It's shorter. And that's how muscle contraction generally works. Okay, so let's talk a little bit about muscle cells. Muscle cells are really, really cool because they're not normal cells, right? Normal cells are microscopic, right? They're tiny, tiny, tiny. They have one nucleus, right? They kind of stay where they're supposed to be, right? Well, muscle cells are insanely long cells. So your muscle cells run the entire length of your muscle. Will one nucleus take care of that long of a cell? Absolutely not. So those cells are fused, and we call those fused cells myoblasts. And so each myoblast contains multiple nuclei, and they share one plasma membrane, right? We call that plasma membrane a sarcolemma, right? Sarcomere, sarcolemma. Inside of each cell or each myoblast are what are called myofibrils. Sarcomeres make up myofibrils. Okay, myofibrils made of sarcomeres. What are the two main proteins in sarcomeres? Myosin and actin. Okay. Now, before we do this, your book doesn't go into very great detail, but I think it's really important that you understand how a muscle cell is organized so that you understand what goes on the inside. So we're going to pretend that this is my bicep. Okay. So if I kind of undo it, we're going to start at the middle and then we're going to build it back. Okay. So here I have one muscle cell that runs the length of my arm, right? This is one long fused cell. It has a plasma membrane, right, that sits on the outside of the cell. Inside of that cell, you can't really see it, it's too far away, but I've stuck a paper clip into it, right? So this little paper clip represents one myofibril. There are many, 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 many myofibrils that are running the length of the cell. And those myofibrils are composed of sarcomeres, repeated lengths of sarcomeres, right? It's like sarcomere, 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 sarcomere. And those sarcomeres are made up of actin and myosin. Okay, so this is one muscle cell. One muscle cell gets together with some other muscle cells, right? We put them all together this way, okay? And we wrap them in a membrane, right? So each layer of this organization has a really tough membrane on the outside of it. Because every time you do this, if you don't have a really tight membrane that keeps everything in place, what's going to happen? Your muscles are going to tear, right? So it's really, really, really important that they're organized and lined up very, very neatly, and then you have multiple membranes around them. So this is called a fascicle. Okay? So a fascicle is a bundle of not myofibrils. Myofibrils are what are coming out of the muscle cell. So it's a muscle cell, which is a myoblast. There are a couple of different names for it, but that is one of them that is correct. What? No, so this whole thing that has multiple myoblasts is a fascicle. So this is one fascicle. So now, to make a muscle, I need multiple fascicles, and usually it's like 10, 12, something like that, but these were all the crayons I had in my desk. So then you take all these different fascicles, and then you wrap them in another membrane, right? Now you take that membrane and you connect that with ligaments, right? To your bones and things like that. And so this is what your muscle really is. Cool? All right. So now I think you're going to understand the pictures a little bit better, I hope. Okay. So this is one myoblast. So what are we looking at here? One crayon, right? One cell. Now inside of that cell, I said you have myofibrils. That's the little paper clip that's sticking out, right? So they've basically taken out one myofibril. Then if we look at that myofibril is divided into sarcomeres. So this, let me do a different color than red. This is one sarcomere. This is one sarcomere. Okay? So it's just sarcomere after sarcomere after sarcomere after sarcomere. So think about that. Sarcomeres are microscopic, right? But if they all shorten, if every sarcomere in every myofibril, in every myoblast, in every fascicle in your entire muscle contract all at the same time, all those teeny, 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 tiny contractions put together end up in making the muscle, right? Okay, so if you look at a sarcomere, right, or you do a cross-section of a myofibril, you'll see things that are really thick and things that are thin. This is why we call them thick and thin filaments. So here in the middle of one sarcomere, right, a sarcomere is from Z disc to Z disc. That's one sarcomere. You're going to have a couple of really important proteins. One of them is the myosin. That's the one that can actually physically move. That is the only protein that can move. Then you have actin. Actin is our thin filaments, right? Actin, myosin. Now, it's great that you got actin and myosin, but that actin has to be, and the myosin has to be anchored to the sarcomere, or just because they slide past one another, you're not going to actually get movement, right? So the actin itself is actually anchored to the Z disc protein. The myosin, on the other hand, has a special protein called titin. Holds it tight, right? Titin. And it's like a spring. So titin can be compressed and be stretched without exerting a force other than anchoring the myosin to the sarcomere. So it's really, really, really important. Okay, so it's going to be this combination of protein interactions that are going to lead to our contraction. What do we need to occur in order to get movement? If you don't eat very much, are you going to be able to exercise a lot? No. What does eating give you? ATP, eventually ATP, right? So all of this movement is going to be ATP driven. Do you remember when we were talking about ion transport? We said that there was something in particular that transported calcium. That was really important. Remember SERCA? SERCA transports calcium from the sarcoplasmic reticulum to the cytosol and vice versa, right? So all of these movements are going to be ATP dependent, and they are going to depend upon calcium concentrations inside of those muscle cells. So there are two big like chemical factors that are going to induce the movement.
Circa is just like a circle.
No, SERCA is the pump. SERC. SERCA is the calcium pump. that's going to induce this, right? Because why do I contract my muscle? I thought about it, right? So I initiated a action potential along my nerves. I thought about it, that was one thing. Then I sent it to my peripheral nervous system, which sent it down my arm, right? And then I have a connection between a nervous cell and a muscle cell. And that electrochemical activation of our muscle cells really eventually ends up activating these calcium pumps. And it's this calcium change that actually induces a conformational change in these binding proteins. Okay, so we said actin and myosin, right? Myosin are our thick filaments, and so they have the head and the tail. The head is what actually grabs onto actin. The tails are what kind of like wrap around each other in order to make that one big thick filament. Actin are individual little bitty monomers that end up lining up in a chain. And that's what makes it, and that's why they're thin, because they're just two monomers side by side that kind of twist as you add them. But they're very thin, very thin. So let me tell you something about actin and myosin. They really like each other. They really, really like each other. If possible, myosin is going to bind to actin. Absolutely going to bind to actin. So we have to have a way to ensure that myosin isn't always interacting with actin. So we have to have, I like to call them bodyguards. Okay. So tropomyosin and troponin are like two bodyguards that are going to tightly associate with actin, and they're going to prevent myosin from binding to them. Okay, so prevents myosin from binding. Okay, so we have these two proteins that are like bodyguards. They protect actin. Now, y'all know everybody can be distracted by something, right? Okay, so these two bodyguards can be distracted by calcium. If calcium levels are high enough, those two binding proteins are going to go, hey, calcium, and they're going to leave the actin. And then myosin can go and bind. Okay. All right, so here's kind of our scheme. So I told you have your actin strands that are your thin filaments. Do you see that you have a troponin complex every so often inside of the actin filament? So that's troponin. And then you have something called tropomyosin, which ends up kind of wrapping all around the actin filaments. It's the tropomyosin that's actually blocking the binding sites of actin from myosin. Okay. And when troponin binds to that calcium, it induces a conformational change, which then exposes those binding sites for myosin to bind to actin. Okay. All right. So your troponin and your tropomyosin, these are our two that if you add Ca2+, right, you're going to get a conformational change. How many times have we talked about this? Something binding induces a conformational change in a protein. All the time, right? Okay, so that conformational change exposes the actin's myosin binding site. So only when calcium levels are high will myosin bind to actin, right? Myosin is waiting. Myosin wants to bind to actin so badly, so badly, but it can't do it unless there's a high enough calcium concentration. And those calcium concentrations are triggered by that electrical impulse that's received by the muscle cell. Okay. So here's kind of our sliding filament model, right? When we have an influx of calcium, that is going to change the conformation of our troponin, our tropomyosin, and allow the myosin to bind to the actin. So calcium 2+, this allows binding. It does not induce a contraction though. What else do you have to have a contraction? ATP. So together, these two things will allow for contraction. We're going to talk about the cycle and how that happens. So if you look in actual muscle tissue, you can actually see this. This dark area here is a Z disc. This is a Z disc. So from here to here is one what? One sarcomere. So from here to here, this is a sarcomere. When you get to the top of the thing, it doesn't like you to write. Okay, so this is one sarcomere. So do you see that there's different colored banding here, where we have this kind of light area between the Z disc and the dark area? We call that light area the I band. So the I band is composed of just actin. In a relaxed muscle, how big is the I band? Small or big? It's pretty big. It's pretty big, because when the cell contracts, these myosin tail groups basically grab onto the actin that are like hooked to either end of the classroom, if the classroom is a cell, right? And pulls them in. So the walls get closer, the Z discs get closer. Well, what happens is that the Z discs actually get closer to the ends of the myosin thick filaments. So if we look here, right, a relaxed cell has an I band that's really big. A contracted cell has an I band that's much shorter. So if we look here, this is our I band. This is the dark part, which contains our myosin tails. So this is the A band. So what kind of a muscle do you think this represents? Is this a contracted muscle or a relaxed muscle? This is contracted. This is absolutely contracted. It's very, very thin. Okay. All right. So now we're going to kind of like zoom in and look at the molecular level. Yeah.
So the I band shrinks whenever it's contracted, but does the A band change?
No, the A band cannot change. The A band is the distance from here to here. It doesn't change the myosin here to here. See? So the A band can't change. The only thing that can change is the I band. The I bands get smaller, which means that the sarcomere overall gets smaller. Yeah. Like, this is so exciting, right? They're like, no. It's so cool though. But actually, I mostly studied non-muscle actin, which is kind of funny. But that's okay. So, right, this is our relaxed state. Myosin heads are not bound to the actin. But do you see these little like circular shaped things that are like empty little cavities inside of the actin? That represents the myosin binding sites. So what's covering those myosin binding sites? Tropomyosin. It's blocking those binding sites. And you have every so often complexes here that are troponin complexes. Those troponin complexes are what bind to the calcium. And when that undergoes a conformational change, it shifts the tropomyosin off of those binding sites. So here comes calcium, right? Calcium induces conformational changes. Now, look at these, they're exposed. As soon as those things are exposed, myosin is binding right away. Myosin's like, yes, right? So excited. All right, so you have to understand like a complete circuit. All right, so can I zoom in on this? I don't even know that I can. I'm sure that there's a way, but it's okay. All right, so when we start, right, we have our actin, we have our binding sites that are covered, and we have a molecule of myosin. Now, that myosin at this point in the cycle has an ADP and an inorganic phosphate. What does that tell you? An ATP was there, but it was already hydrolyzed, right? Okay, so calcium influx. Calcium comes in, induces our conformational changes, exposes our binding sites. Myosin is like, woohoo, yeah, right? Okay, so it binds to the actin. Once that binding occurs, that triggers that inorganic phosphate to be released. So our inorganic phosphate comes off. And this is really cool because you can monitor activity of actin-myosin interactions just by monitoring amount of inorganic phosphate released. It's a cool little readout. Okay, so once that inorganic phosphate is released, this is what induces the power stroke. The inorganic phosphate release triggers the power stroke. Everybody wants to think it's hydrolysis of ATP that causes the power stroke, but it's not. It's actually the conformational change that comes right after release of that inorganic phosphate. So when that released inorganic phosphate occurs, this is the ratchet. This is when you pull, that myosin pulls the actin in. Okay? Once it's completed its power stroke, it then releases the ADP. So now it doesn't have anything bound to it, but it's still hooked to the actin molecule. Once a new ATP comes in and it binds to that new ATP, it can let go of the actin. But it's not in the right conformation, right? It's still ratchet. It's still like in. So it has to reset. So when you have that ATP in the binding socket and that ATP gets hydrolyzed, that hydrolysis allows it to reset and be ready to go. So this, they call that the recovery conformation. Now, if calcium's still present, what happens? It's going to reach for a new binding site, ratchet again, and keep going and keep going and keep going. What's going to stop that cycle from happening? What's that? When all the calcium's gone, but where's the calcium going to go? How are we going to get rid of it? Circa is going to basically turn on and it's going to pump. So if you want it off, if you want to stop, right? Circa will turn on and pump Ca2+ from cytosol back to the sarcoplasmic reticulum, right? So now we're going up a concentration gradient. We have lots of calcium in the cytosol. So we need ATP energy to pump that calcium back in the sarcoplasmic reticulum. Then our bodyguards are not going to be distracted anymore, and they're going to recover those actin binding sites. It's a pretty cool, it's a really cool mechanism. So you want to see an animation? Yeah, right? Awesome. Okay. Let's see if the volume. I try not to blast y'all out, but then it takes a second. Okay, and turn this off.
Most proteins, most notably the cytoskeletal protein actin and the actin-based protein myosin, must work in concert to power muscle contraction. Muscle cells contain numerous myofibrils, which consist of interlocking thick and thin filaments. The thick filaments are made up of myosin protein subunits, and the thin filaments contain actin, tropomyosin, and troponin proteins. Z-disc proteins serve as anchors to the thick and thin filaments. The protein titin functions as a flexible spring connecting myosin proteins to the Z-disc. According to the sliding filament model, thick and thin filaments slide past one another to shorten the distance between adjacent Z-discs during muscle contraction. In relaxed muscle, myosin binding sites on actin are blocked by tropomyosin and the troponin complex. Muscle contraction is triggered by the release of calcium ions from the sarcoplasmic reticulum. In the presence of calcium ions, troponin undergoes a conformational change that reveals the myosin binding sites on actin, allowing the myosin head domain, which is bound to ATP and inorganic phosphate, to bind tightly to the actin thin filament. When the myosin head releases inorganic phosphate, it undergoes a large conformational change called the power stroke that pulls the active fiber along the myosin fiber. ADP is then released from the myosin head. After a new ATP molecule binds to the myosin head, myosin disengages from the active filaments. ATP hydrolysis returns the myosin head back to its starting conformation, allowing the cycle to continue. Muscle relaxation occurs when calcium levels in the cytosol decrease as a result of active transport of calcium ions back into the cytoplasmic reticulum. When calcium ion levels decrease, the myosin binding sites on actin are blocked again by tropomyosin and complement, returning the cycle to its original elongated state. The sliding filament model elegantly describes how force can be made at a molecular level to affect change at the level of cells and tissues.
Isn't that really cool? Like these teeny tiny changes all add up to how we move our bodies. I mean, it's just awesome. It's awesome. But did you notice how the length from here to here never changed? What changed was the I band, right? From here to here. So when we talk about the banding patterns, it only has to do with like taking a picture of it under a microscope, right? You have a mixture of proteins. Just because you're in the I band, do you only have actin in the I band? No, there's actin in the I band and part of the M band, right? And as the muscle contracts, now we have more actin overlapping the myosin, right? So I know it's a little bit confusing in learning the banding and learning the proteins, but it's a way to monitor, like we can measure, we can put a muscle cell under a microscope, change conditions and actually measure the banding pattern changes and know what's going on.
So since Titan is stretched from the A band to the Z disc,
Yep.
Titan also be considered part of the I band?
Yes, yes, absolutely, absolutely. Yep. And Titan's really cool because you can have mutations in Titan that lead to muscle instability, right? Because if Titan doesn't work, when your muscles are contracting and you're not anchoring myosin to the Z disc, your muscles will fail and you will have skeletal muscle disorders. So they're all super important. Mutations in any of them lead to massive skeletal disorders. Like, so exciting, right? So awesome. Like, not really, but okay. All right, y'all ready to do something different? Not my favorite thing ever, but guess we'll do enzyme mechanisms, you know. All right, so enzymes are super important. Maybe not as important as active. No, they are. So think about, let's take a general example, right? If somebody has high cholesterol, what do we prescribe them? Anybody know? Statins. Statins are a class of drug that we prescribe to patients who have high cholesterol. Why? What do they do? Like, this is why it's awesome to be a biochemist, because you know about all the different drugs and how they work and what they do, and every single one of your family members calls you and says, so my doctor prescribed me this new drug. What does it do? All the time. So you have high cholesterol. Your doctor says you need to take this daily statin drug. What is it doing? Well, there are two ways that you can get high cholesterol. Number one is what you eat, right? And number two is your body makes cholesterol. Cholesterol is very important. It's a very important signaling molecule. If you don't have cholesterol, you are not going to be telling your body what to do and when to do it, because you won't have the precursor molecules that are required. They're also a very important component inside of cell membranes, right? So you need cholesterol. But if you're eating a ton of cholesterol, do you need to make it? No. So that's what statins do. Statins are inhibitors of cholesterol synthesis. So they're inhibitors of specific enzymes within the pathway for cholesterol synthesis. So if you're going to eat a lot of cholesterol, we're going to stop your body from making cholesterol. That's why a lot of times, if you can reduce your cholesterol levels, then you can go off the medication, right? This is why everybody should know about biochemistry. Everybody, right? Okay, so when we talk about enzymes, if you understand how the enzymes work, then you can start to design drugs that say, oh, we're going to inhibit this. We're going to change that, right? Or we're going to inhibit this particular cholesterol synthesis enzyme, but not that one, right? You can get really specific. So you have to understand how enzymes work, the amino acid residue side chains that participate in the interactions. It actually gets, this chapter gets like super detailed. I know that's a scary thought, right? What? It's only 2 chapters on this test. So we were laughing about it just a few minutes ago, right? But tonight, go study. Please, please go study. All right, so we're gonna do a general enzyme overview. We'll talk about the structure-function relationship, reaction mechanisms. Reaction mechanisms they're a challenge, and enzyme kinetics are as well. So those are going to be the 2 probably most difficult sections, 3 and 4, and then regulation. All right, so before we start talking about enzymes, tell me important characteristics of enzymes. In order to be an enzyme, what?
Lower activation.
You're going to lower the activation energy of a reaction.
Or faster. You get the product faster.
You make the rate of your reaction is sped up, but it doesn't change the equilibrium concentrations of your reactants and products. That is super important, super, super important. And then the last thing, there's one more thing, that enzymes are not changed by the reactions that they catalyze, right? They're reused over and over and over and over and over again. And they can be both proteins and RNA, right? Okay, so these 2 gentlemen, James Sumner and John Northup, these are the 2 guys who got the Nobel Prize for characterizing and purifying the very first enzymes. Pretty cool. Urease was that very first one, but enzyme purification is even more challenging than normal protein purifications, right? Because not only do you have to preserve the structure, you also have to preserve the enzymatic activity of that protein. So it gets even more detailed. So when we talk about enzymes, they are so insanely specific. Like, I can't say that enough. And we used to think that it was like a lock and key. How many times have you heard that? Lock and key, right? In biology, it's the lock and key model. We don't believe in that model anymore, but it's easy to explain. And so that's why it's still used. So in the lock and key model, what was thought was that our substrate fits into a particular hole inside of the enzyme, and it fits perfectly, right? And so the substrate doesn't change, the enzyme doesn't change, right? The structure doesn't change until you convert that substrate into product. It's the only change that happens. Really? No, no. Everything that I've taught you so far this semester has been how dynamic proteins are, right? How they change, how they're constantly altering their conformations and all this stuff, right? So inherently in your minds, you should be going, red flag, no way, right? So what we believe now is the induced fit model. So in the induced fit model, it is an analogy, I use this analogy of a baseball glove fitting on your hand. So is the baseball glove flexible? Absolutely. Is your hand flexible? Absolutely. Okay. So the enzyme has to be able to accommodate basically an ill-fitting substrate. The substrate is not in the perfectly optimal conformation to fit into the enzyme. It almost kind of doesn't make sense. But what you really want to happen is you want to induce conversion into that really high energy intermediate, right? And you want to make that conversion really, really easy. So you want to promote that conformational change into your high energy intermediate. If you perfectly fit the substrate, would you want to go to the intermediate stage? It's going to stay as the reactant. So what's the point, right? That's why this makes perfect sense when you really start to think about it, right? And so it allows for a certain number of weak interactions between that substrate and the enzyme so that you basically get into the site. So you're in, but then you need to do that conversion to the high energy intermediate and then eventually to the product. And so you get tighter and better interactions going to the intermediate. And then when we get to the product, do we really want to hang out with the product? No. So it's going to be fewer interactions when we get to the product. And then that's why the enzyme releases the product. Okay. All right. So when we talk about where a substrate fits into an enzyme, what we're talking about is the active site or the binding pocket. It's the same exact thing. So here's an example of phosphoglycerate kinase. I don't know if I wrote that. I don't think I did. This is phosphoglycerate kinase. What does this enzyme do? It's a kinase. It phosphorylates. What does it phosphorylate? Phosphoglycerate. Like it's not, they've actually done this pretty well. They've actually named these things nicely, I have to say. That's all it does. So your substrate is what's going to fit into the pocket. What is going to interact inside of this active site with that substrate? Do you see these things that are hanging out into the pocket? What are those things? What is that? They're side chains. They're amino acid side chains. That is what is going to facilitate the interaction between the substrate and the enzyme, right? And so when your substrate binds, it's these residue side chain interactions that promote our catalytic activity. And so you have to think about there being an environment inside of the cell. That's one general area, right? But what can be really, really different is the extreme local environment of that enzymatic pocket. If it's buried deep within the protein, right? Maybe it's excluding water. Maybe we have a lot of positively charged side chains in there, right? There are all kinds of different things that could be altered within the enzymatic pocket that allow for a unique environment and therefore optimal conditions for the catalytic activity to occur, right? Because you got to think, most reactions, if you're in the lab and you want to speed up a reaction, what do you do to it? You optimize it. One thing that you can do is increase the temperature, right? Most chemical reactions occur faster at a higher temperature. Can you do that in a cell? No. So we have to look at other conditions that we can change inside the enzymatic pocket to promote those changes to occur, right? Without altering temperature, without altering pressure, right? We're very restricted within the cell. Okay, so here's another important characteristic is that once that substrate binds, you're going to induce a conformational change in the enzyme. And this is what we have been talking about for chapters now, right? So this is our example. This is hexokinase. Hexokinase is a kinase, right? And it does what? It adds a phosphate group to This one you got to kind of think about. Our substrate's glucose. Why do we call it a hexokinase? Because it's a hexose. It's right. It's just numbering the number of carbons, right? So this can phosphorylate carbons, molecules with a certain number of carbons, right? Okay, so this is an extreme example. Most enzymes do not undergo this huge of a conformational change, but it's very easy to see, so it's a good example. So do you see how normally when it's unbound, we have this enzymatic pocket right in here. It's exposed to water. Well, it's exposed to the solvent. That's good, because what's inside the solvent? It's substrate. But once the substrate binds, now in order to facilitate that kinase reaction to occur, now those two basic sides of the molecule kind of collapse in and hide the glucose from water. And that actually helps to promote the phosphorylation of it. So a lot of times what you're going to see are conformational changes that promote the reaction. Okay. All right, but usually they're not that big. I mean, that's like an insanely large conformational change. Okay. All right, but I did the same thing. Okay. But how do we get this? How do we know? Like, okay, these are really pretty pictures. How do we get these really pretty pictures? What is it? What did you say? Yeah, the structures. You're going to do crystallization, right? X-ray crystallography. You're going to look at the structures, and you're going to look at the structures with substrate over here, and you're going to look at the structures without substrate. And then you basically overlap the two, and then you'll get where those each individual amino acid is moving, right? So when we look at this, we're going to really zoom into that catalytic pocket and look at how minuscule changes in our side chains actually have insanely large effects on the molecule. So you can look at a space filling model and you can look at a ribbon diagram, right? So space filling models look at what? Well, but space, what fills space? An atom is mostly empty space. So what gives you the borders of an atom? Electron clouds. So it basically shows you electron clouds. But then sometimes it's more important, well, it's more informative to look at our secondary structures, our beta sheets, our alpha helices, because a lot of times those secondary elements will undergo changes. Like, remember when we talked about the F helix moving, right? So it's easy to see that, oh, these residues are in one alpha helix. And it's this alpha helix that moves that induces the change between the two subunits, right? So what you could see when they crystallized this is that they were able to see water molecules inside the catalytic pocket here, but they never saw water molecules inside the catalytic pocket once the glucose substrate was bound inside of the enzyme. Okay. All right, so another thing that's really important when we talk about enzymatic function, just because you have an enzyme, do you want it to work? Not all the time, not all the time. You want it to be on call for when you need it, but you don't want it to constantly work. That is not going to be beneficial for the cell, because you have processes that are degradation and synthesis of the same molecules that have to happen in the same cells. So if all of your enzymes were active all the time, you would be futile cycling through things all the time. And it would be a complete and total waste of energy. So we need to have ways to regulate enzymatic activity in cells without changing things like temperature and pressure and all that kinds of stuff. So how can we do that? There are two major ways that we can do that. One of them is bioavailability. What is bioavailability?
What she's going to say, so like take a drug sample or something like
What you can attain, amount, right? Amount of your enzyme. How do you change the amount of your enzyme in your cell? It's a protein, right? How do we get proteins? We're going to start with DNA, we turn them into RNA, and then we make proteins, right? So that control is multifold, right? So how do we control mRNA expression? Not post-translational, that's protein modifications. You're gonna change your ability to transcribe the gene from the genome, right? So you're gonna look at, we'll say mRNA expression, right? This is through transcription factors. Now that's great. That's how much protein we're making. If I make one molecule of hexokinase, is it there forever? No, no, no. So protein turnover is another way that we can change bioavailability. We can also put our proteins in particular places inside of the cell, right? So cellular location. Because you can have one biosynthesis process occur in the cytoplasm, and the degradation would have to occur, let's say, in the mitochondria, right? And so it would allow you a way to separate those enzymes. Okay, then you can alter catalytic efficiency. When we talk about catalytic efficiency, what's catalytic efficiency? How fast, how well the enzyme works. So how can we modify that? We've talked about a couple of different ways that we could do this so far. Change something on it, right? What are we going to change on it? What's the most common? Phosphorylation. What can we phosphorylate? Serine, threonine, and tyrosine. It's an R, tyrosine. Okay, so that's one thing that we can do. So when we do this phosphorylation, right, we're changing, what are we really changing? We're changing the 3D conformation of the protein. So in order to get that three-dimensional conformational change, then we phosphorylate it. There are other ways that we can do that though. We can have non-covalent modifications. So we can interact with certain molecules inside of the cell that will induce a conformational change. It's an interaction, but it's non-covalent, like AMP, for example, right? A lot of processes that require ATP are inhibited by large amounts of AMP. Why? If you have a lot of ATP in the cell, what did you just do? I just ate, right? I got lots of energy. What happens if I have a high level of AMP? I'm struggling. I don't have ATP. So do I want to do the enzymatic processes that require ATP? No, I want to shut them down, right? So some modifications make the activity increase. Some modifications make the activity decrease. But they're not, like, just because you phosphorylate something doesn't mean it's going to become more active. Phosphorylation could be either way, right? Binding could be either way. It depends. All right, y'all are telling me it's time. It's time. We'll finish this next time. So remind me We did phosphorylation and we did non-covalent interactions. Okay. All right. I'm trying. I'm working. I've gotten all the multiple choice and I'm working on the discussion. So I'm doing the best I can, I promise. Yes. What's that?
Yeah.
Some people did really great and some people did not.
I feel like I'm one of the people that did not.
Don't say that. Don't say that. You ready, Christian? Christian, yes. Okay. See you later.
It's like maple.
Did y'all like my model? Did y'all like my
Yes.
muscle
I
model?
did. I thought it was really helpful. I
Yeah?
really did. I'm gonna think about that on the test.
Okay, awesome.
You were holding it
I
right
like
there.
You're gonna think of me. Oh.
I'm just, if I don't know what it is, I'm just gonna contract.
Just contract your muscle and it's gonna come to you.
Oh, it gets shorter.
Why'd you come up? That chair up here. How you doing? Yeah. You had a chance to study? Yeah. So this is how we start to present it. You have a germline mutation. If that mutation occurs in the basically developing embryo, right? That mutation moves into germ cell and it moves into cytosol. Right? Because at one time you are a one cell organism, right? And so if in that one cell, the DNA mutated, every cell is going to get that mutation. So now you have a mutation that is germline and is also expressed in the and so you would physically display the symptoms of like and you can also pass that on to your children. But like for me, right, like, okay, I have normal DNA, I divide a few times, now the cells up here that code for my head, right, they got a mutation that said, make sure you make blood vessels, right, don't. Well, then I'm gonna get a birth mark, but because Christian, you okay?